Ladies and gentlemen, welcome to Quotidiano TV on our today's talk show in Compass with Professor Maja Ravnikar from Ljubljana, Slovenia. Maja, welcome to Compass. How are you doing?
Thank you for the invitation and good day to everybody. I am fine.
Thanks for joining us on such an interesting and also unusual topic. I must admit that very few scientists are familiar with the tobacco mosaic virus, with toboma viruses, and particularly with the way these viruses might spread via water. Maja, tell us something, please. How did you come across this topic to investigate how viruses, which we usually believe not to be able to survive outside a cell, may well endure in water?
Well, it's a history a little bit. I need to tell a little bit of history. It was around year 2000 when we had a company in Slovenia and this company produced special filters which can concentrate molecules, proteins, and I said, why not viruses? Because I worked with viruses 20 years before. And on the other hand, at that time, we also worked with a virus, potato virus Y, which was a big epidemic that was virus infecting potato. And it was obvious that the virus is very stable. So if we put the sap of the plant and test it after 10 days, it was still there. And in this time, it was obvious that viruses and their RNA is not so unstable that we thought before. And I looked at the old literature. It was Professor Koenig from Braunschweig, from Germany, who started a work on viruses in water. And she used centrifugation and infection of test plants to see whether the viruses in water could be infective. And actually, she found positive result. Also, some scientists in Hungary and former Yugoslavia also repeat these experiments positively. And at that time, the technology was not so good as it is now. Therefore, this research stopped. But I was very interested in that and used these special filters for filtration of viruses to concentrate the viruses from the water. And actually, there was a lot of viruses. And not just this possibility of concentration, because we know in the water the concentration of viruses is really low, but also molecular techniques which were developed, like PCR-based technologies, which we know all after COVID, qPCR and other techniques, enable us to see lower concentration of virus in the water. And this is how the journey starts. First with concentration, then with detection. And after 10 years, a new technology came. This is high-throughput sequencing, which enable us to identify every DNA or RNA which is present in the water. It's not dependent whether this is virus, bacteria, fungi, plant, animal, whatever. So we can see what was in the water before. So we are seeing the history of the water.
What determined me to invite you is, first of all, you are a professor at the National Institute of Biology in Ljubljana in Slovenia, and you've published a paper 11 years ago with your colleague, your co-author, Professor Natasha Mele, also from the Center of Excellence of Biosensors, Instrumentation and Process Control, also in Slovenia, and you published a paper, Plant Viruses in Aqueous Environment, Survival, Water-Mediated Transmission and Detection. And in this paper, which is remarkably interesting, you also mention how former Yugoslavia scientists have detected in the 80s also the tobacco mosaic virus in the Danube and in the Sava River. What do you think might have determined them to investigate a virus which is, for many, many scientists, almost unknown?
Well, actually, for plant virologists, it's very famous virus. It's the first virus which was found and seen under the electron microscope. Why? Because this virus is extremely robust. It can stay in an environment for months. And it can also stay in tobacco leaves after they are dried and produced for cigarettes. So this is, it was a fortune to see this virus first and did an investigation. Also, the crystal structure of this virus was first described between all viruses. So that's why for virologists, it's famous virus. And because it is so stable, it is easy to find him because it will stay in water or also in soil, different environments for many months.
And in water, if I'm not mistaken, I think there you said that it may stay up to 3,000 days, which,
yes, right.
Which is like, let's say, 7 to 8 years.
Yes,
it is extremely long.
It is extremely long because the virus is really stable. We also know for some bacteria and fungi, let's say fungi spores, that they can stay alive for many, many years. Of course, you can detect the virus after this long time, but let's say for survival of the virus, you need to do experiments every time again, because it depends on the sample you test.
But crystallized, in case it gets crystallized, is there a time limit at all, or crystallized it may stay, let's say, forever?
Well, if you have, in the case that you purify the virus, that you try to concentrate it from the sample, then the virus will form crystals. So they are prepared from the virus particles, which can very nicely align one to the other. And this is what we are calling the virus crystal form. And well, actually, in this form, it's stable like it is without crystal form.
Is this the same crystal Stanley had crystallized it and received, I think, 1946, the Nobel Prize, or because he received the prize for crystallizing the tobacco mosaic virus? Is this the same crystal? Or
yes, it is the same crystal. They try to purify, and in this case, viruses will come together, line together. What actually happened also in the cell, in the plant cell? We can see this kind of crystals also in plant cell.
Do other plant viruses also produce these crystals, or are these crystals something typical for the tobacco mosaic virus?
Also, other plant viruses which have this rod shape of particles.
So similar viruses.
Similar, but also spherical viruses. Icosahedral viruses can also shape a crystal, which is different, but again, viruses are coming together, and because they have this very nice shape, they can make a crystal.
And how may such a crystal, understand that such a crystal is the result of a concentration process, how may this crystal be then again dissolved? Stanley had proven that he could crystallize it 12 times, dissolve it, and that the tobacco mosaic virus maintained its toxicity, although it changed the structure, became a crystal.
Well, actually, the viruses usually do not change the structure. Okay. They just go apart. And this you can do with changing the pH of the solution.
It sounds very easy.
Yeah, because they are actually, we have on the surface of the viruses, there's a protein usually. It could also be a lipid membrane, but usually for the viruses which can do crystallization, we have proteins. And of course, proteins work very different with very different pH in the solution.
Mm-hmm. Particularly in this paper, which I was referring to there, you explain also the mechanism how plant viruses, I mean, the tobacco mosaic virus, including are water mediated, how this transport may work. Why is it important to look at this aspect of transport, and how easy or how difficult it is to investigate the tobacco mosaic virus in water?
Well, for tobacco viruses and also tomato mosaic virus, it's not very difficult to find it. In addition, we have pepper mild mottle virus. This one is again tobacco virus, which is spread all over. You can also find it in the guts of human because it can go through the gut system, and it's not then activated because it is so stable. So it's not difficult to find it and detect it. And actually, in last years, we investigated more water samples, water, rivers, lakes, wastewater, and found a lot of viruses, which can also be infective still there.
And tell me, do the concentrations of the tobacco mosaic virus detected in water vary considerably in the course of time, or they are more or less the same? Have you observed any declining or increasing concentrations since you started your investigations? What could you tell us, please?
Yes, for those viruses, we were not especially interested in these changes of concentration, but actually we can see them all the time. What is not the truth from some other viruses which are less stable. And let's say what we did, we observed the gastroenteritic viruses in wastewater for the whole year. And we can see, let's say, for rotaviruses and noroviruses, for which we know they are causing the problems, gastroenteritic problems, they vary in dependence on the epidemic. So you can somehow see what is the situation with the virus in the population. When you are looking at the wastewater, you will see how many virus is circulating in the population, whether this is population of human or animal.
So your research, which I've been conducting for decades, I believe was extremely useful for your country also in detecting coronaviruses in water. Is that right? What could you tell us, please, about how your research was extremely useful and helpful in detecting this completely different or maybe similar pathogen agent?
Yeah, actually, our research started because we have an interest how water can influence plant production, and if we have infective viruses which can infect plants after they are going through, let's say, wastewater treatment plant, what we actually shown and published in 2020. It is important that you are checking the water which you are using, especially if the water is circulating, let's say, in greenhouse system, because in this case you can transmit the virus. And there was our research which shown that this is true, that we can, in the hydroponic system, if the infected plants are releasing the virus into the water, and if this water is coming in the contact with healthy plant, the plants will become infected. Of course, this is not very fast procedure and can take, let's say, few months, but when the plant is infected, it goes very fast around. And because we had all methodology to concentrate viruses, to detect viruses, also to do the isolate, search for different isolate of virus when COVID came, and it was obvious that it is possible to see the virus in the wastewater, or we try to see whether we can see it in the wastewater. We, in one month after the starting of close down, we start to do experiments and try to see the virus in wastewater. At that time, that was in 2020, March and April, the concentrations in Slovenia were quite quite low. So, but anyway, we spiked the virus and we saw that it can survive and we can detect the virus. So actually in the next few months, we prepare the country monitoring state monitoring for SARS-CoV-2 in wastewater, and we tested 7 places in Slovenia and detect virus twice per week. And also, in addition, we developed a method that we can detect different isolates of virus. So we can search for these varieties in the genome changes, and can search for a new mutations and new isolates. And that we did until this March. And then the whole system went for our national laboratory for human health and environment, because we are a research institution and we do another research. This was especially important because when you are testing the wastewater, you can see not only the concentration of viruses which are there for the patients with symptoms, but you can also see the patients which are not ill yet, or they don't show the symptoms. Especially is this important when you change, let's say, the system of detection, you introduce new method, or you stop to do very intense detection of patients. So this, I believe, this system is not important just for SARS-CoV-2 virus, but also for new emerging viruses. Let's say we very fast develop the system for some other viruses for detection, because the methodology is more or less the same.
So it's basically an add-value system to a previously detection system for plant pathogens, since you have all the instruments and all the laboratory infrastructure available.
Yes.
Was not such a big step to implementing this alert detecting system for coronaviruses. Maja, what could you please tell us, what exactly, in case of COVID, gets detected in the water? Is it, are your tests referring to the spike protein, or looking at proteins of the spike, or looking at the nucleic acid? Look, I'm not a scientist, I'm just asking. So
the question
what are the tests actually looking for?
Yeah, actually, when we are developing the method, we use the whole particles, but then detection was based on RNA of virus, and we used qPCR method, which is the method detecting the RNA and multiplying the RNA, of course, that you can see then the signal. So and it's the same for isolates, different isolates of virus. Of course, it's very difficult to combine a new isolates in a wastewater where you have a lot of different parts of RNA. It's much easier to do it in patients, but we succeeded to do it in water. So this is extra plus.
Yeah, congratulations. So you actually are looking for the RNA for the virus and not just for the spike.
Yes.
Because I think I read some papers, and they were referring rather to the spike, which itself may induce this toxicity of the virus. So could you actually detect both the spike and the RNA? Are these different tests, or I understood something wrong, and it's solely a test for RNA in case of water?
Yes, of course. These are two very different tests, because a spike is a protein, while RNA, nucleic acid. And in this case, to detect RNA is much better, because the proteins are not so stable. Let's say SARS-CoV-2 virus is much less stable than viruses, which we talked before. Therefore, the concentration of the proteins is much lower. That's why RNA detection, and the whole world is doing that now, is much more representative. The system is different. It could be ELISA test. Actually, the system which is detecting the proteins, and in the case of wastewater, this is very dirty sample, and can inhibit also the enzymatic reaction, which help you to detect proteins, spike proteins.
But in case of the way how coronaviruses behave in water, I mean, you're a plant pathologist, you're not a virologist in human viruses, but still, if I may ask you, which are the similarities and which are the differences between the way, let's say, the coronavirus and the tobacco mosaic virus do behave in water in terms of let's say, the water purity, the pH, in terms of temperature, in terms of hygienic conditions, is wastewater something maybe the tobacco mosaic virus would rather prefer than clean water? And also other similarities or differences which I might have not referred to.
Well, it's a lot of similarities, especially, let's say, if we compare tomato mosaic virus with rotaviruses or noroviruses. They are all very robust, and it's known that both can survive wastewater treatment, especially because wastewater in general are not focused on microbiological threats, but more on chemical, organic cleaning. On the other hand, of course, tomato mosaic virus is much more stable than SARS-CoV-2 virus who is referring to COVID-19, and will stay there. And it will not be so important what is the temperature, what is the pH, what is the surrounding, whether the water is clean or not, while SARS-CoV-2 is much more sensitive. So we don't see that a wastewater which contains SARS-CoV-2 is more problematic than usual wastewater, because there are also not just viruses, but also bacteria, human bacteria, which can infect people. That's why it is somehow similar, but on the other hand, it is different. We need to check for every virus.
Yeah. Another aspect which I'd like to ask you is, now we're talking about water, detecting such unusual viruses in water. While we're talking about water, we also need to refer to fog and rain, since fog and rain, which get produced by the condensation of water, may mediate the transport of such pathogen agents. And you are also in your article referring to this mechanism, which, well, that was for me something extremely surprising, to see that viruses may get spread or may get mediated by fog and rain.
Well, actually, they were found in fog and rain, and there were no real experiment which can confirm that just rain can, with rain, you can infect the plant. With hydroponic, yes, with irrigation as well, but not with the rain, because this is very difficult experiment to do. But it is true that the viruses, and especially plant viruses, were found not just in plants, but also in water, in soil, in rain, also in ice. So they are really could be found everywhere. This is not surprising, because we know now that, let's say, the liter of seawater contain 10 to 11 virus particles. They are the most abundant agents in the world. But as I said, not more than 10 to 15 years, we have the tools that we can do research on very different samples and see this beauty and diversity of viruses.
But what do these units in water refer to? Is this the concentration of the virus in seawater?
Well, I'm not sure whether I got your question.
No, you mentioned 10 to 11.
Aha, okay. So these are viruses in seawater, and these viruses are infecting the plankton, more or less plankton in seawater, and they are contributing to the CO2 which is going out of the water very, very much, because the plankton will, because of the viruses, take apart, will and then of course multiply and so on. It's a very, very important factor in the organic matter and also atmosphere that we are reading.
Yeah, you are quoting in your paper Castello, in another paper from 1995, and there he describes how the virus virions were detected in clouds and fog.
Right, right.
And having done a bit of research on that, look, I'm not a scientist, I found that even in the cloud and fog building process depends on the ice nucleation. And that the tobacco mosaic virus has, in comparison to other nanoparticles of mineral origin, the nucleation temperature at somewhere around -4 degrees, whereas other mineral particles have a nucleation around -30. And these scientists describe how theoretically the tobacco mosaic virus may produce this ice nucleation in clouds and thus produce rain.
Yeah, this is not surprising because actually the protein of virus is attaching the water. So virus always have this water coat around, which you can of course not see, let's say, under electron microscope, but that's true. So that's why it somehow attack water molecules.
And given, for instance, this tobacco mosaic virus, just purely speculative, has such an extreme resistance on the surface, could it then in water, resistance in water, could it then in a higher concentration, since water may easier condensate from the sea surface, or let's say from a lake, than it may on the soil, could it then create spontaneous clouds and rain?
This is very difficult for me to answer.
I know. It's
but
a pure speculative question. I mean, it's
what I would like to emphasize that one thing is to find the virus, and the other thing is whether it will infect the host. Because we know very well for plant viruses that you need to have a specific concentration of viruses that can infect the host, and in addition you need to have wounds on the plant, or in the yes, micro lesions, or in the root. And it means let's say and on the other hand, we know that if the plant is growing in a soil where the water is contaminated, the roots will concentrate the virus on the surface of roots. And because roots are growing all the time, these micro lesions are appearing there all the time. That's why virus can enter the plant.
But the funny part is several scientists, because we've spoken before about COVID, they published papers about the COVID weather. And they looked exactly at the meteorological conditions at the onset of the pandemic. And this topic became to me interesting since I read another paper written by an Iranian expert in the history of medicine, and he describes how the influenza in Iran started with a cold rain in one evening, and then later with a mild wind, and basically most people were sick the next day. And fun enough, there's a paper, a study written by professors Moore, Loveluk, Clifford, Korontiki, McCronick, and so further from Harvard and Maine University, an article from 2020 called "The Impact of a 6-Year Climate Anomaly on the Spanish Flu Pandemic." And they explain a 6-year period of alternative rain and drought, but particularly cold rains during also the summer months. And, you know, in English, you say what walks like a duck, quacks like a duck, and flies like a duck must be a duck. We are complaining about weather anomalies nowadays, which, let's say, could be similar. We say it's Earth climate warming, but it was a similar weather anomaly during a disease which is compared by neurologists, again, with COVID. And we have also a link between the tobacco mosaic virus and COVID. Now, the very speculative question is, might the tobacco mosaic virus at a certain extent create these weather anomalies now, but not in nature? Let's give some scientists completely mentally derailed might have put some high concentrations of tobacco mosaic virus into seawater to create these anomalies, as they were proven to have been during the Spanish flu. This weather anomaly started 1916 and they lasted for 6 years.
Okay, so this is for me, speculation. And let's say if I go back to COVID, we know that we are more susceptible to virus infections in the time when the temperatures are cold, and it is because of our immune system is more susceptible. And so it's very difficult. I don't know any real experiments which can confirm what you said.
Absolutely.
But for sure, the influence of viruses in environment and in our, let's say, illness, whatever, is underestimated, and we don't have enough research. So all this data needs to be verified, and the results needs to be done with experiments. If I can prepare one example.
Please.
We let's say hypothesize that viruses can infect plants via water, but we need to prove that, and it was very difficult to prepare nice experiments where you can show that the viruses went to the water. That was not difficult, but then that these particular viruses are infecting the healthy plants. Because if you work with viruses which are stable, you can have them on the hair, on the fingers, on your equipment. So it's very, very important how you prepare experiment. And I know an article which is stating that another virus, pepper mild mottle virus, which is everywhere, can be multiplied in human gut. I saw that article and I don't believe it. Although I am sure that the virus is going through the gut without problem and going out in wastewater and is there. And actually, with these names, kind of pepper mild mottle virus, tomato mosaic virus, this is because when we gave these names to the viruses, we knew one host. That was, let's say, tomato or tobacco. But it is true that viruses can infect 100, 200, 300 different hosts, and that you can find tobacco mosaic virus in tomato, let's say, and vice versa, and cucumber mosaic virus in many, many, many different species. That's why it's very complicated. But yeah, we are in the very, very interesting area with development of high-throughput sequencing. For virologists, it's for me, it's one of the most important inventions in this century.
In your paper, you also say that there are a few examples how even in metropolitan areas or in the Arctic Circle, in Greenland, the tobacco mosaic virus was found, and that the spread to some forests, for instance, given I understood correctly what you have written, is because of it might have been mediated by rain.
Well, it is possible, or let's say for ice cycle, but actually to find viruses in town, this is not surprising anymore for us, because of course we know that viruses can come there through the fruit that you can put into the water through wastewater where it's not really well maintained. Of course, they can come from plants which are growing in the towns, which is normal. So this is not very special now. What is more interesting is that you can find viruses which hosts we know from our region in Alaska. What can tell you that we don't know all hosts of the virus, and that actually with the wind, with the clouds and water in the clouds, viruses can circulate around the globe. But this is what we can see.
What could you tell us, please, about ways or strategies, first of all, to prevent plant viruses from spreading with water, and in case they have spread in water, to decontaminate such water and to eliminate these plant viruses from water?
Yeah, well, it's a good question, because actually when plants are infected, there's no way how to eliminate viruses from plants. What is true, let's say for some fungi, again, for bacteria, it's not true. It's very different in in plants than in human, where we can treat ourselves with antibiotics, let's say, while we cannot treat plants, because the whole composition, how the plant is growing, how it's developing, it's very different. So we really need to take care that we are not using the contaminated water. And here there are some systems, and usually the ozone treatment is used, which is okay, but not very good for water which is more dirty, let's say, because ozone will not come into the particles, inside the particles, and viruses in organic matter will be hidden in this way. Another possibility is chlorination. This is very effective indeed. So let's say
the traditional chlorination.
The traditional chlorination, let's say what they used in the swimming pools. It's very effective, because we try to find viruses, rotaviruses in swimming pools. No way. The chlorination will disinfect the water.
And COVID?
Well, of course.
And
COVID
tobacco
as well.
mosaic virus?
As well. Yes.
So
this is very, yeah, it is. But what is minus? Minus is that you will get a lot of different substances which can be toxic. So
there's no gain without pain.
Yes,
but
always.
Tell me, is your knowledge regarding the transport of viruses in water something always keeping you busy? Look, I'm having a glass of water. Do you often give a thought what water you're drinking when you enjoy a glass of water?
No, we did a lot of testing of drinking water in Slovenia and never found viruses. Only when it was, let's say, floods. And of course, at that time, water was not supposed to be drink directly.
Of course.
Otherwise, we have this plus, or we are so happy that our water is, I never thought about that. It's very, very, very clean. But you asked me about the decontamination of infected water. And here we are working with together with our colleagues from other institutions. We are working here with a non-chemical way of disinfection. Cavitation is one. Cavitation is mechanical way how to destroy the viruses. We published mean? It means that you push the water through a very, very tiny place, and when the pressure is very high, on the other side, bulbs are formed with very, very high pressure and high temperature, and at that time bacteria and viruses will be destroyed. Well, actually, cavitation is a phenomenon which somehow destroy propellers in the water. if you use propellers for a long time, they will be injured. And it is because cavitation of this physical phenomenon. And we use this also in preparation of system which can help with the disinfection of water. And in other system is plasma technology. So plasma, let's say, is formed when you have storm. And we coupled together plasma technology with this cavitation machine and getting disinfection of viruses very, very fast.
You know, we are saying in Romania traditionally that once water had passed 7 stones, it's clean. So I guess that this physical process, I tend to believe, of cavitation may be somehow in nature be encountered when, let's say, in a river, for instance, it will pass literally 7 stones.
We said in Slovenia the same, and I believe it's true. And we also did research on that.
Oh, really?
Yes, we tried to compare the water which is coming from the ground with water in the river, let's say. And the concentration of the viruses is much, much, much lower. But here, when the water is going through the soil and through the stone, the adhesion is coming into the force. So actually, the viruses tend to stick to the organic matter, to every matter actually, and that's why in the end water is clean. So this is a good point for viruses and bacteria, not so much for all chemicals, but for microorganisms, yes.
And last but not least, is such a procedure also helpful for the decontamination of residual water in case of COVID? Some scientists are worried that COVID had reached our seas and oceans and might then also infect other fish, particularly like dolphins or whales. Is that a subject of interest for scientists, how to prevent in future at least that COVID viruses may reach our seas and oceans by water purification plants, not filtering it properly?
Well, I believe that water which is going through wastewater plant system, it's safe regarding COVID, but it's not safe regarding other microorganisms. And that's why it's very, very important that we keep our planet clean and that we clean our water which we are using and elute into the sea. So I would say that wastewater, if it's not cleaned properly, it's a big risk for animals, and not just animals. We are seeing animals, but in the ocean there's so many microorganisms, many animals and plants which are very important for the big animals in the end, for the ecosystem. And this is according to me the main problem. And this decline of biodiversity which we can see here is because we don't see our the result of our lives and what we do. We can see just if there is no more elephants or I don't know wolves and so on, and dolphins, but it's much more complicated. Let's say we see big decline of insects, big decline of birds, and this is important. A decline of bees, let's say. This is very, very important.
It's a very fragile ecosystem. And I think as long as we don't know, as you mentioned before, how such plant viruses may, at the end of the day, truly proliferate and infect other species, let's say by water, I don't know if it's not too early to find the guilt. Of course, we all need a healthy and clean environment. But given the tobacco mosaic virus or the tomato mosaic virus may produce rain and weather anomalies, it could be also an instrument to point out the finger at people and saying, oh, listen, it's your fault, it's your guilt, all the consequences in nature. It could be an easily instrumentalized and manipulative pathogen agent to reach some goals one of us may be not familiar with. We cannot exclude it, as far as I understood in our discussion now.
I believe that many people do not understand that viruses are all around. And let's say bacteriophages, viruses which are infecting the bacteria, are very, very stable as well. So I would not put tomato mosaic virus as one very important virus. It's one of many, many viruses which can go into the air, into the clouds, into the water, and so on. Because we have viruses all around. And actually every day we eat billions of viruses in plants, in water, and let's say also in water. We have bacteriophages and so on, which does not cause any problem. On the other hand, of course, they can change our microbiome in guts. So the things are very complicated and very interesting.
Professor Ravnikar, thank you very, very much for having joined us here on Quotidiano TV today. Our discussion encompassed about water-mediated plant viruses and how your research was extremely helpful in helping us to better combat COVID, since your system is useful for detecting this virus in wastewater. Thank you very much for having joined us, Professor.
Thank you very much for the invitation.
Have a nice day and greetings to Slovenia.
Thank you.
Bye-bye.
Bye-bye.